marks the end of Tammy's 4 years in the PhD portion of her MD PhD degree in my lab. And once you see the talk, I think you'll agree that what she's completed in this period of time is actually quite remarkable. So during her tenure as a graduate student, her promise and talents as a researcher have been recognized by a number of awards, including a University of Utah Graduate Research Fellowship, and also a prestigious F31 training grant, which is awarded directly from NIH. This is not one of the training grants we have on campus here. So in preparing this introduction, I thought quite a bit about what it is that makes Tammy so effective and successful. And I decided if I had to come up with one word that summarizes Tammy's essence, as it were, the word would have to be fearless. And that's fearless both in her science and fearless in her approach to life in general. And when I say fearless in science, what I mean by that is that if there's something to be done, whether or not it's something that she's done before, Tammy has no pause. She basically dives right in, learns what she needs to learn, finds the people who know how to do what she wants to do, and gets started. And so because we're a biochemistry department, in biochemistry or enzymology terms, we'd have to say that Tammy has little to no activation energy barrier. The word fearless is also appropriate because Tammy was the first person in my lab to move from single-celled organisms, because for years we've used yeast as a model, to genetics and disease pathology in mice. And so this means that in addition to intellectually directing the project and doing most of the experiments, she also was instrumental in setting up the necessary infrastructure to do this work. So as a consequence, I can honestly say that I have learned as much from Tammy, if not more, than I hope she's learned from me. And I can also say that she's actually ready to start her own laboratory as a result of this experience. So in short, it's really been a great deal of fun to have Tammy in the lab and to see this project develop. And with that, I want to turn the podium over to her. The title of her seminar is Moving Mitochondria, Mitochondrial Rho GTPase Miro1 Protects Against Neurodegeneration.
It's my turn now. Okay. So first, I'd just like to say thank you so much for coming out here. All of your support means a great deal to me. I've met many people along the way on this project. So, first, I'd like to draw your attention to the focus of the first slide. What you have here is a time-lapse video of a mouse cortical neuron expressing mitochondrial targeted GFP protein. As you can appreciate, mitochondria in this time-lapse video is moving in two directions. Either the anterograde direction from the cell body here, down the axon to the synaptic terminal, or we also have a population of mitochondria moving from the synaptic terminal down to the axon, back to the cell body in what we call the retrograde direction. There's also a population of mitochondria that are stationary throughout the whole time-lapse period. So this to me suggests that mitochondrial movement is highly regulated and dynamic. They can go in two different directions and their speeds vary. And when I first started in Janet's lab, this image of moving mitochondria really appealed to me and intrigued me. I wanted to understand why these things move, how they move, and in particular, how does this relate to human disease? So with that, I'd like to introduce my talk, Moving Mitochondria, Mitochondrial Rho GTPase Miro1 Protects Against Neurodegeneration. So as you can appreciate from the previous slide, neurons are highly specialized and polarized cells. And in this cartoon, what shows what we know is that in neurons at the cell body is where the majority of organelle networks, such as the ER and mitochondria, resides. Also, it's where the majority of the biosynthesis occur. And because this is where organelles and biosynthesis occur, there needs to be a way to distribute organelles and biosubstrates packaged into vesicles to other parts of the neuron, such as the axon or the synapse. And to accommodate this, the neurons have developed a very elaborate and intricate microtubule tract highway system within the axons, depicted here. And you can see mitochondria being moved from the cell body to the synapse, and also back from the synapse to the cell body, where they're thought to be needed to turn over damaged mitochondria. As you can imagine, the distance from the cell body to the synapse can vary, and in humans, this distance could be up to 1 meter long. So you can imagine that the integrity and efficiency of this axonal highway system is incredibly important to the cell integrity. And in fact, we know this is true because there are a number of neurodegenerative diseases that is associated with axonal transport defects. So here in the short list of neurodegenerative diseases, I wanted to highlight a subset of these diseases caused by genetic mutations, either directly or indirectly affecting axonal transport. For example, in hereditary spastic paraplegia, it's caused by a mutation in a molecular motor that's required to move cargo within this axon highway. Versus other mutations in diseases like Huntington's, Alzheimer's, ALS, and Parkinson's are directly caused protein aggregates, which are thought to clog up this axonal highway system. And what I want to bring your attention to is, although all of these mutations are caused by different genetic causes, they all have one thing in common. They all have well-documented defects in mitochondrial movement, suggesting that mitochondrial movement may be a step in neurodegenerative disease progression. And with that observation, I wanted to ask whether or not defects in mitochondrial movement has a direct role in neurodegenerative disease progression. And we have some indication that this might be true because we know that neurons are highly metabolic cells and also require ion flux. In mitochondria, delivery is needed for ATP and calcium buffering in neurons. So in this cartoon here of a neuron, you see mitochondria cluster at dendritic spines where they provide energy for sodium, potassium, ATPase, and sodium channels. We also have documented evidence that mitochondria are found along the axons. And here's a lobe of an axon where they are clusters at the nodes of Ranvier, which is highly concentrated of sodium, potassium, ATPase, and sodium channel. We also know that mitochondria tend to cluster at the synaptic terminal, where they again provide energy for synaptic vesicle release during neurotransmission and also buffer calcium during neuron activation. However, the role of mitochondrial movement in neurodegenerative disease progression has never been directly tested. So I just wanted to ask a very simple question. Is a defect in mitochondrial movement sufficient to cause neurodegenerative disease? So to answer this question, I need to learn more about what the molecular features are that distinguish mitochondrial movement from other types of cargo movements in the cell. And what we know is here you have a cartoon of a mitochondria. It expresses a highly conserved outer mitochondrial membrane protein called Miro1. Miro1 binds to a mitochondrial specific adapter protein, which bridges its connection to a motor protein called kinesin. And together, this mitochondrial movement complex moves mitochondria along microtubule tracks. What's important to note about Miro1 that it's conserved from yeast to human, and it has a very unique protein structure in which it has 2 GTPase domains flanking 2 calcium binding EF hands. Now, because Miro1 is a mitochondrial specific protein and it has multi-protein regulatory domains, we thought that this would be a good target to focus on to study mitochondrial movement specifically. We know that Miro1 is ubiquitously expressed based on work done in Pontus Aspenström's lab. This is a northern blot showing Miro1 transcript levels at different human tissues. And as you can see, Miro1 is expressed in regions that are rich with mitochondria, such as the heart and skeletal muscle. And we also see transcript expression in the brain. So our approach to target Miro1 was to first generate a Miro1 conditional allele. And really with the help of Janet and Jared, who designed the targeting construct, they designed 2 LOXP sites flanking exon 2, which encodes the Miro1 GTPase 1 domain, which we show in our lab in yeast is required for Miro function. So by targeting this exon 2 domain, this construct was electroporated into embryonic stem cells by Susan Tomowski, and I screened for stem cells harboring this Miro1 engineered LOXP allele by Southern blot. And you can see here in these 3 examples of ES cell clones that Miro1 LOXP migrates at a lower band, and that's because they're using the probe and the strategy cutting with HinD3 and E81. Any allele harboring the Miro1 LOXP will be a shorter band versus the wild type would be 2 HinD sites. So these embryonic stem cells were then used to generate Miro1 LOX-P mice. Miro1 LOX-P mice were exposed to Cre recombinase to generate the Miro1 knockout allele. Miro1 knockout allele was verified by genomic PCR, shown in this plot here. And during this time, I also generated polyclonal rabbit antibodies against the mouse Miro1 protein, and a postdoc in our lab, Agnieszka Lewandowska, showed that there's no Miro1 protein expression in the Miro1 knockout. So when I generated the complete Miro1 knockout animals, what I noticed is that they appear cyanotic at the day of birth, and 100% of them that I recovered died, and this was 28 animals from 125 embryos collected. I know that these animals are alive the day before birth at E18.5 because with the help of Dr. Kevin Whitehead and Candace Carter at the ultrasound core, we were able to do fetal ultrasound and show that the Miro1 knockout animals actually had beating hearts before delivery, which is surprising looking at these Miro1 knockout, because earlier I showed you the transcript of Miro1 is ubiquitously expressed. So you would think a protein that is ubiquitously expressed would have such a much more dramatic phenotype. And instead you see well-developed head structure and limbs. At this point, I wanted to know why these animals are dying. So I did a post-mortem autopsy and saw that the lungs were not expanded. So what I did is harvest the lungs from animals at E18.5, which is the day before birth, looked at tissue morphology based on hematoxylin eosin staining, and saw that all three genotypes had relatively normal lung morphology. And this was also verified by vet pathologists that we worked with at ARUP, Dr. Lawrence McGill. However, when you looked at animals at the day of birth, which is P0, wild type and heterozygous animal have air within their lungs, and you can see the air spaces here in white, essentially expanding the alveolar tissue. You do not see this in the knockout animal, suggesting that these animals died because of a respiratory failure. So what are the possible causes of respiratory failure? Either these animals have a primary lung defect, or they have a nervous system control defect that's required for respiration. We ruled out a primary lung defect based on the HB staining I showed you earlier, where we didn't recognize any gross lung tissue morphology defect. So focusing specifically on the neural respiratory control pathway, we know that inputs from the cerebral cortex can give signals for breathing. There's also specific motor neuron clusters called nuclei, which I'll talk about a little later, located in the brainstem that's required to innervate muscles required for breathing. We also know that there are specific motor neurons in the cervical spinal region that give branch to the phrenic nerve, which innervates the diaphragm, which is the primary muscle that's required for inspiration. So looking at the schematic pathway, I worked my way down and to find if I could find a neuronal defect in the Miro1 knockout mice. I didn't find any histological defects in the cerebral cortex, so I moved down to the brainstem. And in collaboration with Alex O and Ava Feldman's group at the University of Michigan, we saw that Miro1 knockout animals did not properly form two cranial nuclei. So these are clusters of motor neuron cell bodies. We visualized neurons by Nissl staining, which you see in green, and they're also stained with DAPI, which stands for DNA, in blue. So here, one nucleus I will talk about is the nucleus ambiguus, and it's boxed in for you here. And you see the cluster of motor neurons here in the wild type. It's also present in the head, but it's absent in the knockout. And this was validated in 6 to 8 animals. And this is an important finding because the nucleus ambiguus gives rise to motor fibers that innervate the laryngeal muscles in your neck, which is required for breathing. In addition to missing nucleus ambiguus, we also found malformation of the facial nucleus. And the facial nucleus is indicated here in this rectangle dashed box. It's present in the wild type in the head, but you do not see a well-organized facial nucleus in the knockout. So now we have 2 missing motor neuron nuclei in the Miro1 knockout. And this is I want to highlight how surprisingly specific this phenotype is because we looked at other cranial nuclei and they were completely normal in the knockout. So moving down in our pathway, I was curious whether or not there was further downstream effects, like if there was defects in the cervical spinal region, if we had sufficient number of motor neurons that are required to give rise to the phrenic nerve. So again, in collaboration with Alex, we looked at motor neurons at the cervical spinal region using an antibody against cholinacyltransferase. So here, motor neurons are shown as red. And you see, as representative images shown here, the knockout appears to have a fewer number of motor neurons. And this was also quantified in about 6 to 8 animals, and we saw a roughly 50% decrease in motor neurons, suggesting that Miro1 is required for the maintenance of motor neuron. Those motor neurons, a subset of them will give rise to the phrenic nerve, which I mentioned earlier is the main nerve that's required to innervate the diaphragm for inspiration. So when we looked further downstream at the phrenic nerve, we saw that Miro1 knockout animals had fewer axonal branching. So here we looked at neuromuscular junction with bone marrow toxin in red, and we didn't see any obvious difference in the number of neuromuscular junctions. However, when we looked at the phrenic nerve by neurofilament staining, we see a much less robust axonal branching or connection in the knockout than when compared to wild type. And this suggests either Miro1 is needed for neuronal branching to make connections with the muscle, or somehow they're dying back and retracting. So from the experiments I've showed you so far, we concluded that Miro1 is required for the development and maintenance of specific motor neurons that's necessary for breathing. However, because these animals die at such an early age, I was unable to answer my first question, which is mitochondrial movement required is a part of the neurodegeneration and disease progression? So to address that question, I modified my original question a bit and asked, does neurodegeneration happen when Miro1 is lost from postmitotic neurons later in development? And to answer this question, I made a neuron-specific knockout of Miro1 using the neuron-specific enolase Cree driver. So this enolase Cree driver, I chose this specifically because I didn't see any expression of Cree in the brain stems, which will hopefully bypass the breathing issue that I had of the complete knockout. And to check for Cree expression, I used a reporter mouse with Rosa Laxi and did XDL staining, and show you that here, Cree is expressed in the cortex, which are blue cells indicated by the asterisk, and you also see Cree expression in the hippocampus. I also saw Cree expression in the gray matter of the spinal cord, and also in the bone, the vertebral column. It's important to note that there's no Cree expression apparent in the white matter of the spinal cord or in the muscle. So this was a good candidate to use to make a neuron-specific knockout. And so basically, whenever you see a blue cell, it indicates it's expressing Cree, and if it's expressing Cree, that's where Miro1 will be knocked out. So generating the Miro1 knockout mice, I saw that these mice develop symptoms that are very similar to human motor neuron disease. So the earliest neurological sign that I saw was at around P14, so 14 days of age. These mice, when checked for reflex, here in a control, when you lift up the tail of a mouse, its hind limb will splay out. But in the Miro1 neuron-specific knockout, which from now on I'll refer to as NKO, it has a neurological sign where they have hind limb clasping. So this is a sign of neurodegeneration and used for other neurodegenerative mouse models. As these mice mature, they develop additional neurological symptoms. You see here a very pronounced stiff tail, curvature of the spine, which we call kyphosis, a stiff hind limb, also indicative of hind limb spasticity. And unlike its littermate control, it's no longer to distribute its weight evenly on all 4 limbs. Most of the weight now is hunched over to the front limbs. I also noticed that these animals are about 40 to 60% smaller than their littermate control, and I showed that they have a failure to thrive phenotype. If I follow the Miro1 NKO mice over time, as shown here in the gray line, they fail to gain any weight versus their control litter mates gained weight over time. And doing a composite phenotyping assay, which basically scores the neurological phenotypes I mentioned up here in a degree of severity of 1 to 3, 3 being the worst. I saw that the Miro1 NKO here in gray, symptoms gets worse as they age, whereas the wild type controls do not show any neurological symptoms at all. So to highlight, give you a better visual of what's going on with these mice, I'd like to show you some videos. So here's a wild type. You see as it's walking, it's evenly distributing its weight on all four, and the spine straightens when it walks. At 4 weeks old, this is a litter mate control. This is a control, litter mate control of this mice here. You can see a pronounced stiff tail. You can see hind limb spasticity. It's having a hard time coordinating its hind limb to walk. You can also see a kyphosis here. And you can definitely tell that this mouse is much smaller than the littermate control shown above. And at times you do see sort of a mild tremor that occurs in this mouse. And as he gets kind of going, he moves a little bit better. I've also documented that later on, when these animals are a little bit more mature at 6.5 weeks, their motor neuron defects appear to be more pronounced. So here you can see the tail is absolutely stiff at this point. The hind limb is much more spastic. I've also documented that these, and as you can tell from the video, these mice probably don't last very long, and in fact, they do have a shortened lifespan. The Miro1 NKO animals, which is shown in the black line here, typically die between 30 to 40 days of age, versus their littermate control, which is the red and gray line, do fine around this time point. Around the same time, I also made another Miro1 conditional knockout using a motor neuron-specific Cree driver. So this is MNX1 Cree or HB9 Cree. This Cree driver specifically expresses a mature motor neuron in the spinal cord, so not the brain, which is very different from the driver that I used for the Miro1 NKO. And I documented that Miro1 motor neuron knockout do not have any of the phenotypes that I observed in the Miro1 NKO. And based on that observation, I came up with a pathway that suggests the defects that we observe in the Miro1 NKO probably comes from neurons that originate in the cerebral cortex that signals to the brainstem down to the lumbar spinal region to innervate the hind limb. So again, I'm going to work systematically down this pathway to see what we can find is causing the symptoms in our motor neuron, in our Miro1 NKO mice. So focusing on the cerebral cortex, I isolated primary cortical neurons and measured mitochondrial movement using time-lapse imaging. So here are representative images from a wild-type and Miro1 knockout cortical neuron. And these cortical neurons are expressing mitochondrial targeted GMP. This is very similar to the video that I showed you at the beginning of the talk. The quality of movement of these mitochondria during the time-lapse period can be depicted in a chymograph shown here. So in this chymograph records data from, at this corner here is time 0 and distance 0, and over time in the y-axis we can follow the movement of each one of these mitochondria. So for example, if this is our anterograde direction, any line moving in this direction here shows you that the mitochondria is moving in anterograde. Any line moving in this direction, which points to retrograde, suggests that that mitochondria during that time moved in the retrograde direction. Thick vertical lines that you see indicate mitochondria that are stationary throughout the whole time period. So after generating a series of these chymograph data, I was able to analyze both quantitatively and qualitatively mitochondrial movement in cortical neurons. So what we found was when you compare Miro1 knockout mitochondria with wild type, there's no statistical significant difference between the percentage of mitochondria that are moving. However, out of the subset of these moving mitochondria, I found that there was a significant difference in the time that Miro1 knockout mitochondria spend in motion. In other words, they don't, when they're moving, the knockout mitochondria, the knockouts do not stay moving that long. In addition, I found when I looked at the average number of reversals, how often does a mitochondria change direction, we saw that the Miro1 knockout mitochondria change directions less frequently than a wild type. And what that tells us is that perhaps there is directionality problems in the Miro1 knockout, and also the processivity of the mitochondrial movement may be decreased in the Miro1 knockout. So when I looked quantitatively at the velocity of Miro1 knockout mitochondria, I saw that if we only looked at the anterograde direction, the velocity does not appear to be statistically significant. There's no difference between the knockout and the wild type. And this is reflected also in the run length, the distance that the mitochondria travel, when you look at knockout mitochondria compared to wild type. There's no statistical significance. However, when I looked at the retrograde direction, I do see a dramatic decrease in retrograde velocity in the knockout mitochondria compared to wild type. And this is also reflected in the distance the mitochondria travel in the retrograde direction. So essentially what I'm showing you here is that you have mitochondria in the cell body, and in wild type cells, they're moving down the axons of the synaptic terminal in the anterograde direction without any defects. There's going to be a population of mitochondria labeled in red that are deemed to go back in the retrograde direction and perhaps fuse back to the mitochondrial network in the cell body. In the knockout, there appears to be a defect in this retrograde transport, so there's fewer red mitochondria moving back to the cell body. If this model holds true, then you can imagine if I did a cross-section of the axon, there should be fewer number of mitochondria in the knockout compared to the wild type. And I decided to look at axonal mitochondria at the lumbar spinal region, since it was downstream from my pathway. If a defect in the neurons are sending signal down to the spinal column, I should see fewer mitochondria in the axons in this region. And in fact, based on EM, I do see fewer mitochondria in axons from Miro1 NKO animals. So here is a representative image of electron microscopy done at the lumbar spinal region. Axons are depicted here. You can see the thick myelination around axons. And mitochondria are indicated for you by the arrowheads here within the axon. So I quantified over 500 axons per genotype and saw that roughly 40% reduction in the number of axons with mitochondria in the Miro1 NKO animals. So that data suggests that this model holds true, and if this is true, why is a defect in retrograde mitochondrial transport causing such a dramatic animal phenotype in the Miro1 NKO? One possibility is that there may be a respiratory defect in these animals, since earlier that list of neurodegenerative disease I showed you all have mitochondrial respiratory defect. However, based on EM, when I looked at mitochondria within the axons of NKO animals, I saw well-organized cristae formation, which usually when you have mitochondria that lose respiratory activity, they do not form well-organized cristae. So based on this image, it suggests that because of the intact cristae formation in the NKO animals, there might not be a respiratory defect. But however, to test this more rigorously, I wanted to look at mitochondria in live cells and measure for mitochondrial respiratory activity. And I did this by two different assays. One is to look at membrane potential. So in this panel here, you see Miro1 knockout MEFs expressing mitochondrial targeted GFP. So So all mitochondria will express GFP and be green regardless of membrane potential. I also stain these cells with mitotracker, which is a dye that's dependent on membrane potential. So mitochondria will only take up this dye if they have normal membrane potential. And when I look at the double labeling, virtually all mitochondria in the knockout express the GFP and can take up the membrane-dependent dye in the knockout, similar to the heterozygote and the wild type. I also looked at real-time mitochondrial respiratory function using seahorse and to measure oxygen consumption. And here you see the wild type is in the light gray. When you add different stresses to the mitochondria, it's able to recuperate and consume oxygen consumption at a similar rate when compared to wild type, suggesting that there's no, primary defect in the mitochondria. So if there's not a mitochondrial respiration defect in these neurons, then what is causing the animal phenotype that I see? Why is not moving mitochondria in the retrograde direction causing such a dramatic phenotype? So I went back in histology and looked at the spinal cords of the Miro1 NKO and noticed that both in the brainstem and in the lumbar spinal cord, there are these clusters, abnormal clusters, which we're calling Bunina-like bodies. Bunina bodies are typically found in motor neuron disease patients, most commonly in ALS, and they are protein aggregates of unknown origin. And it's actually used to diagnose the disease. And it's interesting because now I have an animal that has motor neuron disease-like symptoms and also histological evidence that it represents a good model for human motor neuron disease. So to go back to my original question, is a defect in mitochondrial movement sufficient to cause neurodegeneration disease? I hope that I convinced you today that the evidence that I showed that Miro1 is required for the development and maintenance of specific motor neurons, and that loss of Miro1 in postmitotic neurons in mammals causes both behavioral and histological findings that mirror human motor neuron diseases, and that Miro1 is specifically required to control retrograde mitochondrial movement in axons, which is a novel finding that a single gene affects directionality of mitochondrial movement specifically. So with that, I get to go to the fun part. So this project has brought me to many different people all over campus and across the US. And I'd like to say thank you first. Aaron Price and Mike McCaffrey, they helped me with a different yeast project that I worked on in the beginning in Janet's lab. But I like to acknowledge them because they spent really a lot of time teaching me about fluorescent microscopy and really cultivating my love for microscopy. And also our wonderful ongoing collaboration with Alex O and Ava Feldman, the University of Michigan, who've been really key to helping me develop this Miro1 knockout animal story. And I showed you Alex's data in the beginning. Also, Dave Weaver and Yuri Hainovsky at Thomas Jefferson, who helped with mitochondrial calcium data, which I didn't get to show you today. But they are ongoing collaborators and have been great help, and it's been really wonderful talking science with them. And Takumi over in Japan, who provided the Miro1 constructs that helped me rescue my phenotype to know that my knockout by May was in fact real. And then back at Utah. So like the focus of my thesis topic today, I actually moved around quite a bit all over campus. And the first place I'd like to acknowledge is Karen Wilcox's lab, and Karen's here today, which means a lot. They are located out in Research Park in the anti-convulsant drug development program. So Karen has been absolutely wonderful. She's opened her lab to me, introduced me to Jerry, who's this great postdoc that I worked with all last summer to learn how to do primary mouse cortical neurons from. And Karen, please tell Jerry that I learned it so well that I was able to teach someone else in my lab how to do it now. like to thank Dr. Lawrence McGill, who is a vet pathologist at ARA, who really helped us filter and go through the histology early on when I showed up. And I have all these H&Es standing, but I don't know how to read them. He was very helpful with that. And of course, Dane, who's also here, who struggled with me to get that tracking data that I showed you, to show you all those nice bar graphs that literally probably took 3 months to get together. And I also like to thank Osama and Ed, who helped me with the mouse MRI imaging that I did, which I didn't get to show you today, but they were also very instrumental. And Susan Tomowski, who's here, who really helped me learn how to make transgenic animals. I started off, she gave me DNA and I didn't know what to do with it, and really helped me walk through the process on how to set up a southern blot, how to screen. And was very, very patient with me when I asked her probably really naive questions, like how do I get DNA from a mouse tail? And Blake Anderson at ARA, who's been super helpful with histology. Dr. Cheryl Palmer, who's a neuropathologist who actually found those bunina bodies that I mentioned earlier. So she's been a wonderful collaboration and so willing to take time out of her busy clinical schedule to look at my mouse histology with me. And in the Winthrop building, you can tell that my talk is very neuroscience oriented. I'm in the biochemistry department, originally in a yeast lab. I don't know anything about neuroscience, so I really need to solicit a lot of help from the neuroscience department, in particular Megan, who I'd come in and throw ideas at her all the time, and she's always willing to challenge me and ask me, why do you want to do this? Why is this important? And also Jen, the technician in her lab, who helped me a lot too. Rich Dordsky, who is great. He read my first and many, many drafts of my training grant and gave us lots of suggestions on how to make the project better. I'd also like to thank the microscopy core, particularly Mike and Chris, who helped me set up the protocol to do time-lapse imaging in cortical neurons. And I recently learned a lot about EM, working with Nancy and Linda. And a lot of people in this building, actually, in the human genetics building. Kirk, who is here, which I owe many thanks to, learning how to do southern blots. And he took time out of his busy schedule for me to show him scanned images of my blots and tell me what I did wrong and how to systematically figure out, troubleshoot these southern blots. And Anne, who's also here, helped me so much with histology and spent so much time teaching me how to do paraffin sections and H&E staining. And Jamie Soto and the phenotyping core helped me with seahorse a great deal. And of course, Janet Bassett, who's here, has been a wonderful support. Keeps telling me, I can do it, you can get through this. And the ultrasound core, which I mentioned, and Kevin Whitehead and Candace Carter. And also Stefan Pulse, who's my co-mentor on my training grant. He's not here today, he's in Japan, but him and his lab have been super helpful at guiding me through this project. His lab is actually split between biopolymer and here in the human genetics building. And lots of people in my building, the actual lab that I'm supposed to be working in. Majority of these people are on the 5th floor, which is great. And the 5th floor in the Emma Eccles-Jones building is a great place to work. But first, I like to thank the ladies in the biochemistry office who really helped handle all the administrative stuff so I could focus just on the science. In particular, Dawn, when I needed to order reagents last minute, and I did very poor planning and told her I need it tomorrow, and she would get it for me. Warren in the Stillman lab, who knows about all the equipment on the 5th floor, and he's here today to support too. Great collaborators and discussions from the Rudder lab. You guys have been wonderful neighbors. And the Tanton lab, which is where my second bench is. So I'd like to thank Dean for giving me a bench there. And in particular, Arvind, who's a wonderful scientist and a great teacher, and really taught me how to make primary meths and how to do a lot of tissue culture. He's been a tremendous help in this project, especially Jessica, who sits right across the hall from me, who's always telling me that I can do it and I have cool data, which I never believe. And everyone in the Shala, past and present member. In particular, Danielle for helping me manage the mouse colony, which I can't imagine that I ever was able to do any of this before she came. And for handling my not-so-secret type A personality. And Jane, who helped clone the Miro1 knockout constructs. Agnieszka, who's helping on the project right now. And all the past graduate students, Chen, John, and Quinn, who really helped me along, really developed my science and kind of like made sure I had all the controls I needed to do. And past postdocs, Holly and Beth, and in particular Beth, who, if you guys know her, knows incredibly how patient and warm she is, and what a wonderful scientist she is. And next, I'd like to thank my committee member, which I'm going to miss dearly. I kind of wish I have a committee member when I go back to med school to keep track of me. In particular, Janet, I guess I didn't really realize until recently how much of a risk and how much confidence you had in me to take on this project. I spent a lot of money and had a lot of independence on this project. And it's her first mouse project. If I was in her position, I don't think I would have given a grad student this much freedom. So thank you for that. I think it has really helped develop me to become a better scientist. And my committee members have always been held their office door open and had wonderful discussions about what I should do next scientifically and also personal and career-wise. And funding from NINDS and, of course, the MD PhD program. So thank you to Jerry and Dean for letting me into the program. I hope I wasn't a disappointment. Or I guess we have 2 more years to come, so we'll see. And lots of friends I've made in Utah throughout the ways. You guys know who you are. In particular, Jessica and Tien, my little trifecta group, who's been with me since medical school. So I'm really excited to see where we end up in the future. And then my family, of course, who are incredibly supportive and always kind of pushing me to do better and to strive for the best. And they were so supportive, in fact, that they took time out of their busy retirement life to go with me to Portugal to support me at a mitochondrial conference I went to present a poster. I know it was really hard for them to go to Portugal. With that, thank you for listening.
I'm thinking back in one of your earlier slides where you showed Miro1 and where it was in the whole scheme, and I'm trying to understand mechanistically why knocking it out would affect retrograde and not retrograde.
Yeah, I have no idea. Everything in the literature since that experiment suggests that it's possible to control both, and I think it's an extremely exciting new detail that we found. So what I didn't talk about, mammals also express Miro2, which is another homolog. So it's very possible that Miro2 is dictating anterograde movement, and Miro1 is dictating retrograde movement. That's a very attractive model. But up until this point, knocking out Miro1 in animals such as Drosophila, which only has 1 Miro, affects both retrograde and anterograde movement. So it could be that mammals develop 2 Miros to regulate movement or different orientation.
Are there any diseases? Are there any diseases that match the Miro1?
No, and I tried in a very amateurish way to try to look for it, and we talked to Lynn Jordy a little bit, and there's no clear evidence that there's a human disease happening in that area. Yes, thanks.
I was curious about the protein aggregates that accumulate.
Yes.
So what's the implication? Is it that you need retrograde movement to clear these aggregates? And is that because
So that's a really attractive model that's never really been thrown out before. So typically in these neurodegenerative diseases, protein aggregates are everywhere, and they thought, you think that they clog up the system. It's never been shown that disrupting mitochondrial movement leads to protein aggregate. You think that protein aggregates affects mitochondrial movement. So it's very tempting to speculate that perhaps you need mitochondrial movement to degrade or turn over some proteins. So perhaps some proteins that are attacked with degradation hitches a ride with the mitochondria as it's heading back to to the cell body per se to be turned over, and that's why you get these protein aggregates. I think that's a very interesting model. It could tell us a lot about how this neurodegenerative disease progression may occur. An idea that I had was perhaps you start off with protein aggregates, it slows down mitochondrial movement, and you end up getting an amplification effect where you get more protein aggregates because you can't move mitochondria. But
So related to that, then it could be interesting to cross it with the SOD ALS mouse, where you are getting aggregates in the mitochondria. You should have it really exacerbated.
So that was one project that I was thinking about doing, but unfortunately I don't think I have time. We have mice from Stefan Pulse Lab. There's a transgene mutation in spinal muscular atrophy, and it basically makes a protein aggregate. And they developed a distinct phenotype, I think around 6 months of age. So I was hoping to put it in the background of Miro1 and see if I could accelerate the progression of those phenotypes. But I haven't been able to do that. Yes.
Tammy, very nice talk, and congrats on a beautiful body of work. As you were giving this, your acknowledgements, I was saying it takes a village, because it went on and on.
Yes.
It takes a suburb. But I have a couple questions for you. In your germline null, you had a very clear neuronal loss phenotype. In your enolase null, it didn't seem as if you had as much neuronal loss.
I honestly haven't looked that carefully. So I still need to go back, and we need to do like Nissl staining to look for neuronal loss in the specific region. So yeah, I can't say that there's no loss because I actually haven't looked carefully enough.
And so just to follow up on that, what are your thoughts as to the basis of the loss of these neurons? Are they undergoing apoptotic death? Are they undergoing autophagic death? Why is it when you don't have mitochondria that move up and down, do you have any idea or ideas as to what might cause that to the mice?
So an attractive hypothesis I have is that they're undergoing death by cytotoxicity. And Karen is actually helping me with those experiments right now, where we're taking organotypic cultures from mouse brain and basically stimulating the neurons. And when you stimulate neurons, you get a buildup of calcium, and that can cause them to die. And because Miro has those 2 calcium EF binding hands, not being able to move mitochondria to locations to sequester calcium may be a part of maybe a mechanism that causes them to die. Does that answer your question?
Sure, yeah.
Yes, Karen. Do you have data for me?
Actually, we do.
Okay.
It's all set in a file for you. A very lovely job, very nice body of work. You should be very proud. My question is, because the mitochondria can't go back, are you getting an accumulation of mitochondria in the synaptic terminals, and has your EM revealed anything about that, and does it possibly interfere with synaptic transfer?
That would be exactly what my hypothesis is. We haven't organized a protocol to look at synaptic region of these neurons. I would guess that there would be a cluster of mitochondria there at the synaptic end, since they can't go back retrograde to the cell body. I tried doing that with fluorescent microscopy, but the resolution just was not good enough. So I think you're right, EM is the way to go. And yep, another thing on the to-do list.
Can't go back.
Jerry.
Just to follow up on a couple of these questions. So the one hypothesis would be that Miro1 is somehow specifically identifying mitochondria in the synapse that should be retro-transported. And I wonder if you can see that. You know, you have this really nice primary neuron culture where you can track mitochondria, and you could probably, assuming you could do gene transfer into those cells, express a tagged Miro1, a GFP tag or something, and see if it is actually translocating on mitochondria in the reverse direction. And if that's true, it might lead you to wonder what's particular about those mitochondria, whether they're depolarized or...
No, I think that's a really good experiment to do. We always kind of grapple with overexpression constructs and having tag constructs that are functional. One of our collaborator right now, Thomas Jefferson, is doing a similar experiment with the MEFs, and we're doing indirect immunofluorescence with the A-tagged version of Miro, and seeing if those localize specifically to mitochondria that are distributed in a particular location. But yeah, I think that's a good model.
Okay, if there's no more questions, let's thank Tammy. And she now has to be grilled by her committee, but I'm anticipating a positive outcome. So I'd like to invite you all back to the break room on the 5th floor, and somewhere between half an hour, maybe 40 minutes, to join us in a celebration for her successful completion, or signatures at least.
Thank you.